This is a brief description of our novel partwise segmentation method with artificial template. Different modalities for single neuron imaging can provide different levels of detail. For example, mosaic analysis with irrepressible cell marker can be used of outlining the morphology, while confocal laser scanning microscopy yields branching details. Integration across multimodality imaging would provide multiple perspectives for morphology analysis. However, the conventional registration method is not suitable for this problem because it cannot handle severe changes in appearance and the mapping function is arbitrary. On the other hand, we tackle this problem as the partwise segmentation with artificial template. This template is independent from spatial resolutions with predetermined compartments. The partition is generated according to specific context derived from the domain knowledge. Then, we align images by segmenting them accordingly to the template. In this work, we solve this problem using the simultaneous registration and segmentation technique. Our goal is to compute the transformation T that best fits the template Lj to the input volume I, which is Li. Our objective function is formulated using the Markov random field framework. The unipotential is composed of three factors. The first factor is the shape energy. It imposes shape regularization of the transformed compartments based on the local boundary smoothness and area. The second factor is the image energy. We assume compartments follow the piecewise constant intensity model, so this term penalizes intensity dissimilarity between I and Li. The third factor is the centerline location term. It matches the axonal centerlines of target image and template. Pairwise potential W enforced spatially smooth transformation using the diffusion regularization. A method follows the global-to-local approach through the global and local transformations. The T-global transforms the template from our standardized coordinate to Cartesian coordinate. The standardized coordinate system describes template compartments with respect to the axonal centerline, so it is resolution agnostic. Contrary, the Cartesian coordinate system describes spatial relationships between compartments which changes with the data acquisition modality. And the T-local finds the best fitting solution Li to the input data I. It is represented using uniform cubic B-spline free-form deformations to reduce the number of parameters. Our results indicate successful segmentation performance, which is mainly due to the consideration of the structure information in both global and local transformations. The global transformation exploits the neuronal morphology directly by describing the volume with respect to the axonal centerline, while local transformation integrates the centerline location term into the objective function, which makes the MRF framework take into account both compartment shapes and volume structure. Therefore, removing any of these components will reduce the accuracy of partwise segmentation. Here we show one possible application of our simultaneous registration and segmentation method for solving the partwise segmentation problem with artificial template. Neuroscientists hypothesize that the nanometer-scale protein network and micrometer-scale cellular complexity are linked with each other. Our method can be applied to the quantitative analysis for the study of this relationship between these two worlds. In this work, we map the concentration map of the protein-protein interaction in low spatial resolution to the high spatial resolution GFP image. Aligning these two modalities provides the information about the protein interaction at the level of branching details. It enables the local analysis of both dendritic complexity and neuronal functionality altogether. Here we apply our method to align both input volumes with different spatial resolutions to the artificial template. For registration, we first maps the low-resolution concentration map of protein interaction to the artificial template to recover the protein interaction level in the standardized coordinate system. template is partitioned based on the topology with respect to the natural environment, shape, and function. Finally, we apply the inverse function to map the artificial template to the high-resolution GFP image and obtain the protein interaction estimation at high resolution.